Hello, my name is Michelle Landay. The date today is June 5th, 2019. This is my week 5 video, biochemical testing for microbial identifications. This is going to be done through carbohydrate fermentation testing. So carbohydrate fermentation testing is it's done to determine the different byproducts of fermentation. We have our phenol red broth here, and this is helps with acidic. It's for pH. So basically what we're going to be looking for is a different color yellow in these, in our broth tubes here. And we put the Durham tubes inside it here to see if it's a acidic product with or without a gas made, which could be carbon dioxide, hydrogen, a lot of different things. This test can be ran with other tests to help really break down the microbes and to get them into specific groups. So for this assignment, we had to clean up our area, wear a PPE, and I have my fentanyl red broth here. I had my glucose container, but now I have my fructose and my mannitol. I have my nutrient broth here, the S. cerevisiae, and I have my S. epidermidis here. Basically, I've already done the glucose just to get a test run at it. And I've already soaked my Durham tubes in alcohol because they take a little bit to dry. It also takes a little bit to get the alcohol out of them, surprisingly. So what we're going to do is I'm going to walk you through the steps I took for the glucose for the fructose. I labeled them. I have my 1 and 1 and 2 and 2 and 3 and 3. So for this assignment, first we need to take our test tube and we need to sterilize the top of this. Alright, that should be good. And now we're going to take our fructose and we're going to dump half of this into this number 2, if it comes out. Let's try this. Try something else here. All right, one more time. All right, so now that it's in there, finally, I'm going to re-sterilize. Put this down, put my lid back on, and I'm going to shake this until I can get all the fructose off the top of my container and it mixes well. So once I did that, I'm going to put this back down in here. Well, actually I'm not. I'm going to actually hold it for a second. I'm going to drop in my Durham tubes here, and these are going to be used to catch any sort of gas byproduct from these. So we're going to be careful, drop it in, and I've already alcoholed those for a while and dried them off. I'm going to put the lid back on this for a second, and then I'm going to make sure that I grab my S. cerevisiae, which is right here. And then I'm going to sterilize the top of this, because what I'm going to do is use my little pipettes over there to drop 4 drops into my red broth. And I'm going to be very careful not to spill this because these are very full. I'm going to grab my pipette, kick out all the alcohol that's been sitting in. I'm going to shake it out till it's dry. And then I'm going to grab 4, probably a little more than that, but I'm going to try to grab 4 I'm going to drop 4 into here. 4 drops. 1, 2, 3, 4. Put that back in the alcohol. I'm going to re-sterilize for later, which is going to be the whole entire rest of the tubes I have. Put the lid back on. Put that out of the way. I'm going to re-sterilize my red broth. And put it back on here carefully, as carefully as possible. Put my lid back on, and then I'm going to wait 12 hours, maybe 18 hours, to see the results. So a yellow is going to be an acid fermentation. A yellow with air in these tubes here is going to be an acid fermentation with a gas byproduct. And like I said, this is a pretty cool test. I could see them running it to help break it down into different specific groups. But I would almost imagine if you're looking for something else, you would have to run another test on top of this test. All in all, pretty cool experiment. I'm going to finish up the rest of these, and I will post the pictures in my lab assignment. Have a wonderful day.